Thank you very much. Just to say that this is, SNPs means single nucleotide polymorphisms, and these are mutations in the DNA. So as you know, we found some genes that are differentially expressed or occur at higher or lower levels in CFS patients than in normal people. But the trouble is that these, and what we did as well was we used these gene levels to derive groupings of patients which had similar gene levels within that group, and we've called those subtypes. So subtypes do exist in CFS, and we desperately need a means by which to identify them, which is objective. So it's difficult to use the gene expression in order to do this. So the idea that we had was to use these SNPs, or DNA mutations. And the strategy we took was to, well, I'll tell you more about that. Well, these are the 88 genes that we found occurred at abnormal levels in CFS. And when you cluster these, you get these subtypes, and they have significantly different gene expression levels within the main CFS group. And the interesting thing was that these had clinical differences. There were differences in their phenotype and levels of severity. And this was a study we did last year where we extended the numbers and found 8 subtypes instead of 7. And this shows the SF36 scores, the domain scores and the total score for the different subtypes. And you can see that these are significantly different. So the hypothesis in this SNP study was to test the hypothesis that gene expression subtypes can be predicted using SNPs, and that we can develop for use as subtype predictive tests in CFS. Well, I've mentioned the importance of subtypes. So the subjects we used were 108 CFS patients, 17 endogenous depression patients, and 68 normal blood donors. And we used only 17 endogenous depression patients because these were the patients that we used in the gene expression study. And we had to get drug-free depression patients who did not smoke, who did not take antidepressants. And in 2 years of enrolling, we only got 17. So SNPs then. A SNP locus is a position in the DNA genome where we know a single base mutation occurs, and where at one position or one locus there are 2 possible so-called alleles. So in one DNA strand it might wobble from a cytidine to a thymidine. And within the human genome there are millions of SNPs. And these occur in humans and all other species. And we believe this is a significant mechanism of DNA variation. And this plays roles in the inheritance patterns of different diseases, the transcription of these genes into proteins, into messenger RNA, and then proteins. And we've got functional abnormalities that result as well that give rise to various diseases. So this shows one SNP in the NCBI, or National Center for Biotechnology Information, within the SNP database. And this shows the SNP here, and this particular SNP is RS9102, and these are the flanking sequences. This didn't come out very well, but this is the Applied Biosystems software that allows us to see how many SNPs there are within one gene. And within this gene, the gene itself is shown here, there are several hundred SNPs. Only these blue ones come out, and these are the ones that are have validated PCR assays but there are many more in black that didn't show in this slide. So we selected 504 SNPs located within the promoter and coding regions of the 88 CFS-associated genes. And there were between 1 and 8 SNPs per gene. We also looked at 359 SNPs which predict ancestry, and these are referred to as ancestry informative markers. And from this you can predict whether a subject comes from Western Europe, from Asia, etc. And validated all of these for use in the Illumina Golden Gate SNP microarray. So this just shows part of this file where we have the gene on the left, the gene bank number, other means to identify these genes, and then the individual SNPs per gene, which we have here. So the ancestry informative marker SNPs were used to predict ancestry, and CFS-associated gene SNPs were analysed between CFS and normal, CFS depression and normal, and between the 8 CFS subtypes. And for any significantly associated SNPs that we found, we would then analyse for the location in, with respect to the probes of the microarray, the Affymetrix array that we use to identify the gene expression abnormalities in the first place, and then analyse these SNPs for a possible location within the CFS-associated transcription factor binding sites that we also identified in this microarray study. To simplify that a little bit, there is the possibility that we find SNPs associated with CFS that could have played a role in raising the level of some of the genes that we found by interfering with binding for these probes and also location within these transcription factor binding sites. So this shows one particular SNP, an ancestry informative marker SNP, and the data that is on the database, an NCBI database, in terms of ancestry. And these represent different studies and different builds of these studies. And from this information, we can predict the ancestry of a particular individual. So the clinical data of the subjects used in the study is shown here. CFS, endogenous depression, normal blood donors, and the CFS patients had typical prevalences of these symptoms that we use to diagnose the disease and typical scores in the Chalder Fatigue Scale, McGill Pain Questionnaire, SPHERE, SF36, and the Pittsburgh Sleep Quality Index. Oh dear, this didn't come out well either. But this shows hierarchical clustering of the subjects, all of the subjects, with respect to the ancestry informative marker SNPs. And what it shows, you'll have to take my word for it, is that these subjects are generally unrelated to each other. And there was a pair here that were quite similar and unusual because one was depression and one was normal. But with respect to the rest, they were more than 50% unrelated. So we found that 22 SNPs were significantly different in distribution between CFS, depression, and normal, and these are shown here. And 11 of these were different in distribution between CFS and normal. When we looked at the subtypes, we found that 148 SNPs had a significantly different frequency of distribution across the 8 subtypes. And interestingly, these were clustered in particular genes. So these are the subtypes across the top, the SNPs down the side, and this shows basically the top of the table because it's too big to show all of it. And the genes which these SNPs occur within are shown here. So these are 5 different SNPs within the ACTR3 gene, and we can see that subtype F has a different distribution than what would be predicted by chance. Subtype G has a different distribution of these ACAP10 genes, etc. And this clustering of SNPs within these genes occurred right down the table. So we looked to see would these SNPs that we had found possibly, might they have accounted for some of the gene expression abnormalities that we found. And we only found one SNP which occurred within one of the genes, and therefore the answer is this is most unlikely. CFS SNPs occurring within CFS-associated transcription factor binding sites, we found 3, which is quite a minimal possible contribution. And finally, we looked at the SNP profiles across all the subjects of known subtype, and the subtypes are shown here. And this figure is the result of 2 consecutive methods to reduce the complexity in this very complex data set. And basically what it shows is that there is surprising separation of these SNP-derived gene expression subtypes. And you can see here that this is a loose cluster of subtype 6. This is a loose cluster, subtype 5. This one, subtype 8, etc. So in this small analysis of 108 CFS patients, I think this is quite supportive towards using these SNPs to identify gene expression subtypes that we've previously identified. So in summary, we identified 21 SNPs associated with CFS versus depression versus normal, 11 SNPs associated with CFS versus normal, and 148 which were different across the CFS subtypes. And the study provides evidence supporting the use of these for prediction of subtype-specific status in CFS. So just to thank clinical collaborators and Nana Shinosako that did the laboratory work, and EMI Research UK who funded the study. Thank you.